In continuation to the previous lecture, today we will start covalent bonding for enzyme immobilization. Immobilization of enzymes by covalent bonding to activated polymers is a widely used approach. It is often a tedious procedure. It is capable of producing an immobilized enzyme that is firmly bound to its support. The range of polymers and chemical coupling procedure that are used is enormous. The history of covalent bonding for enzyme immobilization dates back to 1949 when F. Michael and J. Edwards used the azide derivatives of carboxymethyl cellulose to immobilize a variety of proteins. Activated cellulose supports continue to be popular due to their inherent advantage of high hydrophilicity, ready availability, potential of for derivatives, and the ease with which cellulose-based polymers can be produced either as a particulate powder or as membranous films. It is often more effective not to build the reactive group into the cellulose itself, but instead to use a chemical bridge between the cellulose and the enzyme molecule. The requirements for such a bridging or linking molecule are that it must be small and that once it has reacted with the support, it must have a further reactive group capable of reacting with the enzyme. An example of such a bridging molecule is glutaraldehyde, which contains two aldehyde groups, one at either end of its CH2 moiety. At neutral pH values, the aldehyde groups will react with free amino groups. Thus, one end of the glutaraldehyde molecule may be attached to the support and the other to the enzyme. Covalently immobilized enzymes are strongly bound to their support, so when the proteins denature, they are difficult to remove. Therefore, it is usual for both the enzyme and the support to be replaced. This may result in higher operational cost compared with adsorption techniques in which the support may be reused. Entrapment. The entrapment of an enzyme can be achieved in a number of ways. First is inclusion within the matrix of a highly cross-linked polymer. Second is the separation from the bulk phase by a semi-permeable microcapsule. Third is dissolution in a distinct non-aqueous phase. An important feature of entrapment techniques is that the enzyme is not in fact attached to anything. Consequently, there are none of the steric problems associated with covalent or adsorption methods, that is, the possibility of the enzyme binding in such a way that its active site is obstructed by a part of the supporting polymer matrix. The example of an enzyme retained in agar is a useful illustration of entrapment. A preferable alternative involves mixing the catalyst with sodium alginate gel and extruding this into a solution of calcium chloride to produce solid calcium alginate particles. This technique has the advantage of not requiring the use of high temperatures. However, although it is a popular activity in teaching laboratories outside that setting, it is generally unsuitable for the immobilization of purified enzymes, as these are often able to leak out of the gel. Entrapment techniques for purified enzymes are more likely to involve retaining the enzyme behind some form of ultrafiltration membrane. Immobilization changes in enzyme properties. It was suggested that immobilization might change the properties of an enzyme to enhance its stability. Initially, it was believed that such enhanced stability resulted from the formation of bonds between the enzyme and the supporting matrix that physically stabilize the structure of the protein. Indeed, there are some published reports that describe this phenomena with regard to the stabilization of proteolytic enzymes, which often exhibit more prolonged activity in the immobilized state. This is most probably explained by the fact that such proteases in free solutions are prone to autodigestion, that is, enzyme molecule cleave the peptide bonds of adjacent enzyme molecules, a process that is largely prevented when they are fixed to a supporting matrix. However, the effect of immobilization are more often due to the supporting matrix changing the microenvironment around the enzyme and or introducing diffusional constraints that modify the activity of the catalyst. Consider, for example, immobilization of the enzyme by adsorption onto a polyanionic negatively charged support such as cellulose. If the substrate is a cation that is positively charged, it will be attracted to the support and thus to the enzyme. In this case, the enzyme might well display higher activity as the substrate concentration in its microenvironment would be higher than that in the surrounding bulk phase. Other cations would also be attracted, and importantly these would include H plus ions. Thus the microenvironment would also be enriched in H plus ions, so the pH surrounding the enzymes would be lower than the pH of the bulk phase. Consequently, the enzyme would also exhibit an altered pH profile compared with that of its soluble counterpart. In addition, the immobilization matrix might act as a barrier to the diffusion of substrates, products and other molecules. For example, if a high enzyme loading was put into a gel particle and this was then immersed in substrate solution, the substrate would diffuse into the gel and rapidly be converted into product. Enzyme molecules entrapped deeper within the gel particle might therefore be inactive simply because they had not received any substrate to work on. That is, all of the substrate was converted to product in the outer layer of the particle. Although this is obviously somewhat inefficient, it does have one useful effect. When over time the enzyme within the system denatures, the loss of activity of the enzymes in the outer part of the particle means that substrate will now diffuse deeper into the particle to reach the previously unused core enzyme molecules. In effect, this inner reserve of enzyme will offset the loss of enzyme activity through denaturation. So, the system will show little or no overall loss of activity. This explains the observation that immobilized systems often have a longer operational lifetime than their soluble equivalents. In addition, it is of interest that enzymes bound to natural cell membranes, that is phospholipid bilayers within living cells, will also probably demonstrate these effects, and immobilized systems thus provide useful models for the study of such membrane bound proteins in living cells.